I'm happy to introduce you to something which we actually just have released at the beginning of this year. And Ivan mentioned it's called Biomolecule toolkit, which sounds pretty generic and actually is meant to be generic. It is somewhat following the footprints what we have started off in chemistry with JChemBase. And it's sort of a kind of successor of JChemBase in the context of that you need some different technology in order to interrogate biomolecules in the same way as we're used to doing that with small molecules. Small molecules we pretty much all understand quite well. You have some sort of visual representation. You have a computer representation which is aimed to store this information in your databases. And if you just want to apply the same tools to a large molecule like an antibody, you'll end up something like this. That can also be expressed in terms of a kind of chemical information graph, but actually it's not very useful. What you rather look into is you try to abstract some of that information into a different layer of information and then try to make sense of this. But whatever you end up with, you have some pretty pictures which some of you will be familiar with, that this might be something like an antibody molecule that's coupled to some chemical entity right next to it. And all of that together makes an antibody drug conjugate. That's not the end of the story though, because this would imply that this is something like a defined exact structure. But in fact, that's a kind of stochastic product where this little chemical entity bound to one of the chains or more chains, if you consider that these red dots are actually representing the same expanded chemical structure, then you'll have some sort of a distribution where a number of payload molecules are bound to your antibody, which is then typically characterized in a number, the antibody to drug ratio, and that can vary. So how do you get that information to be part of your representation? Because that's not real chemical structure anymore. I'd like to somehow give you a feeling for why we've developed this biomolecule toolkit and what we think it should be. First of all, it is something which lets you index and store and query biomolecules with lots of different kind of additional data and properties compared to chemistry in a single toolkit. Everything which relates to the chemistry is pretty much powered by our JChem technology, which is industry-proven and well-established. But in addition, these kind of additional bits of information need to be represented in a standardized fashion. And that's what we've added into the biomolecule toolkit here. What it does for you is it provides you with a layer which helps you to unambiguously define such complex entities. And as such, if you can imagine that you have different groups in the development process of an ADC, some of them are chemists, some of them are biologists. They typically don't understand each other very well. But nevertheless, it will be very advantageous for you if you can have them work on the same kind of data content. The challenge for us is we need to provide them with different views depending on what their primary domain expertise is. And that's what we are trying to solve with this biomolecule toolkit. Last but not least, it's a toolkit. That's kind of the first step in a process towards developing applications which support design, which support data analysis in the domain of biomolecules. So this biomolecule toolkit at the moment is like a puzzle piece in a future larger platform, or if you are interested upfront in kind of early adopting this technology, you can build this into your own applications in-house. It is currently shaped as a web service API, but I'm pretty sure that over time we might actually also release it as a Java API, as we've done this for JChemBase. I'd like to extend now a little bit into the context of registration, what we've just heard by Csaba, and try to somehow highlight some of the things which make biomolecules a little different from regular biologics. Probably the easiest case, which is a one-to-one forward translation, is when you're dealing with plain peptide or nucleic acid sequences, unmodified. It's pretty simple. You can use your established bioinformatics tools, handling sequences as a string of individual characters. That's all pretty neat, but what do you do when it comes to molecules which have somehow biological sequences involved, but then carry chemical modifications or some sort of unnatural chemistry inside? That's where the kind of bioinformatics tool scope typically ends, and that's where the chemistry starts. But still, you don't want to see these things necessarily in a just full chemical representation. So that's where we put in the biomolecule toolkit in order to be able to abstract the sequence information, for example. We go a step further than mentioned. There is some sort of ambiguity in describing an antibody-drug conjugate. The payload binds in a certain ratio to an antibody molecule, which is additional data, which is structurally relevant, but it's not part of the actual structure itself. If we go even further, then we have to make these kind of discriminations, because even if you have the same antibody, you have the same payload, if the ADR is different, your registration system should be able to find that these are, in fact, two different entities. And you might want to set up business rules for that. That's something we support with our biomolecule toolkit. And last but not least, you're often involved in registration of kind of late-stage molecules and development. But use cases in early-stage discovery are typically that you might have little or no information about parts of these molecules. Consider the fact that you're in-licensing an antibody, but you don't get the sequence with it. But you do a conjugation reaction in order to bind it to a given payload, and then you run the assay results. That thing only has the notion of, well, there is an antibody coupled to something else, of which you know the structure, because that's the kind of knowledge you own. You would like to represent this thing, because it's a sample you're working with, right? So it should end up at some point in your registration database. That's something we support with our biomolecule toolkit. Once you've captured all of the relevant definition details of these entities, what you really want to do is you'd also like to be able to search them, because that's the key part of doing analysis. You want to somehow shape queries against the content of what you've just created in order to do data interrogation. So I'm just showing here an example which is coming from our client in a video. This is somehow a rudimentary, very basic web client interface we've built for demo purposes of the toolkit we've built so far. It has a small search interface, and what I'm looking for is I'm looking for a molecule which has a certain search subsequence motif, and then it is myristoylated. So I'm looking for, in fact, this piece of a chemical structure is a long aliphatic chain in order to show that there is a hit which corresponds to specifically this myristoylation to this chemical structure. And it has some, you might not be able to read it, but has the relevant subsequence motif. I think you'll get the slides later on, so maybe you can just review it at home. Another thing which I can demo to you, however, it's not out there on our demo station. You'll have to come and find me because I have it on my laptop only. Campbell recently has released, I think George is here somewhere in the audience. He's over there. They have migrated most of their biotherapeutic, biopeptide content into this new Helm format. And I've just thought, like, okay, how can we actually make a meaningful demo out of this? So actually, we just grabbed their content and loaded something like 20,000 molecules, which are provided now by the Campbell version 20 database in Helm notation, and import this into our toolkit database. And together with it come like 2,800 monomer definitions, which make up the dictionary in order to build these biomolecules. And if you're interested in, and I can show it to you on my laptop, it's pretty cool, actually. This is about the technology side. What I'd like to somehow advertise to you is that we intend to provide this biomolecule toolkit as a platform for a number of different applications. I've just mentioned registration. I have talked a little bit about search, and how does it go into real software tools? Well, first of all, it's coming with the API, which means that all of our other products kind of digesting API calls could potentially use the capabilities of our biomolecule toolkit. Among these are quite a number, but you can also think about third-party applications, not necessarily ChemAxon owned. What I'd like to focus on today is Instant JChem, because we've built a really cool prototype using the biomolecule toolkit technology, and this is what I'm going to show next. Most of you will know that you can create new projects for handling small molecule structures in Instant JChem. Well, we can do better than that, I think. So we create a new project that's connecting to a MySQL database which contains our macromolecule content. And it actually then recognizes as soon as you promote this thing to an entity that this is a kind of new biological entity. And once you then go into the grid view for these macromolecules, you see a table popping up with these large molecules. And that kind of is one of 3 challenges we've modified Instant JChem to. The first thing is that it doesn't connect any longer to a kind of JChem table specifically designed for small molecule structures, but to something which feeds it with large molecule content. Second of all, I won't show this today, but we have also enabled the kind of alternative structure renderer, which uses the structure renderer out of the Helm editor, which is an open source tool. And last but not least, we are also able to perform searches on this. So what you see here is then just an illustration of the fact that you can draw something in our registration clients. On the right-hand side, you have a live connection to the database. And then when you update the schema, once you have defined your molecule, you actually find that over there. So it can be accessible from a number of tools, and you can easily imagine that that's not only something we'd like to see in Instant JChem, but that's really something we'd also like to see in Plexus Suite someday. That's the focus of probably some of our close future developments, in fact. Once you are there, you can think about further expanding what do you do with that data. So you could think about a kind of analysis module in Plexus, which is able to handle biologics, like decomposing the view into this is the antibody part, this is the linker and the drug part of a payload, and then doing chemical interrogations on the chemical parts of that molecule, or doing sequence-based searches on the antibody part, just in order to get that data all into a single intelligent spreadsheet. Another thing which you might want to do is related to structure activity analysis that can feed your design of certain biological sequences. Small peptides, for example, it's a quick way to somehow enumerate say an alanine scan in order to identify hotspots for activity. And then in the second step, once you've found that your third residue of that peptide is actually triggering a cliff in the activity profile, you're going to replace this by a number of natural and unnatural residues which you think are suitable replacements in order to trigger a change in its activity profile. Then you can do all sorts of filterings in order to make sure that you have sequence motifs there which comply with that they don't get degraded by proteinases flying around in the serum or whatever physchem properties you'd like to have your peptide. And then you register these things, make it and measure experimental data based on these. So in a nutshell, this is sort of a preliminary roadmap. I think for the next quarter of the year, we're pretty much focusing on extending this prototype, which we've built in Instant JChem, make sure that it becomes available in Plexus Suite as well. We'd encourage you to get in touch with us if this is something even remotely relevant for you, because we live on your feedback, and we have some plans, but you may have even better thoughts about that. Our plans are good, not so good. We'll see about that. In the second quarter, we'll address probably a pain point which currently arises from how you actually define these molecules. We use a tool which is open source, which is the Helm editor. That's pretty useful as a desktop application. We have made an applet out of this, but as you all know, technology-wise, applets are no longer supported by the end of the year, so we're going to come up with something which is a sort of viewer for these molecules, which provides you with renderings which make sense in different contexts, how biologists or chemists would like to view that data. Maybe later that will culminate in an actual editor in the way as, well, we've seen it with Marvin.js, for example. And then later throughout 2016, we can talk about the itty-titty bits. We can think about how to put the representation capabilities into action for actually providing you with tools for SAR analysis. We'd be happy to actually get that integration running also into JChem for Office, for example, or well, have the editor. That's all from me. If you have any questions, I'm
just wondering what sort of how this carries through into the Java API, for example, or the command line tools like Marvin View. So for like computational chemists, for example, who might be analysing cyclic peptide data, might want to use that within the sort of standard command line type environment rather than, you know, the very nice web-based integration you have here. So how does that work at the moment? I mean, does Mole convert, for example, can I read these into the JChem base?
So what we have right now is kind of a very rudimentary function where we don't put too much emphasis on making this available from the command line. The reason for that, this is becoming more important when you actually look in property calculations, you want to do some batch processing of certain things. Our current entry point with the chemoinformatics stack is in fact a conversion service which allows you to kind of export something into a mole format, and from there you can go wherever you want. As long as you bring results back, we'll be able to somehow pull that in as additional data or as kind of annotation to the entities you're working with. The reverse way is something we're actively developing right now, going from an actual chemical structure file into the biomolecule abstracted view in order to perceive your sequences, what are the peptides hidden inside a chemical graph, for example. We currently see that really more as an enabling technology, which by itself provides you with real basic functionality, much like JChem-base. When we look at tools which actually do work and processing on that data, I think that will come at a later stage.
Are there any limitations on the size of the peptides that you're working with?
Not really. Full antibody, for example, it's like 1,500 approximately amino acid residues if you count both heavy and light chains. And it's no issue at all.
Pretty much a link to the second question is about the analysis. I think you touch upon at the end. Would you think about implementing something where we can cut the things, analyze the segment, for example, in macrocycle, just not viewing or storing, but just to analyze why this macrocycle, this activity, maybe it's because somehow the linker, so some really specific analytical tools that can be directly plugged to this more viewing way, or sub-searching. Do you have something in process about?
Not exactly, but the crucial thing is that when you think about the ADC example, which I have shown, you kind of arbitrarily annotate certain parts of that molecule to have a certain functionality. You know that your antibody is the targeting moiety within that bigger construct. You have a linker which is annotated to be kind of the connecting piece between your warhead and the actual antibody. Being able to annotate this is something which we do. And that is probably key to what you mentioned. So if you're looking at, I don't know, the tether of a macrocycle, for example, and see if the length of the tether has some sort of an impact on the activity, then that's something we should be able to annotate and then analyze accordingly. But that will require probably for each use case a slightly adapted modification of how the tool interprets the annotations.